Proteins Part 1. Now would be a good time to have a look back at your GCSE work, if you still have it, because you will have to already come across proteins and you will already have some information about what they're composed of, and in particular the good sources of proteins. Some of the sources are shown on this picture here. If we look at the structure of proteins to start with, we'll have a look at what they're mainly composed of, which elements are most commonly found in them. And like most of these molecules, they are rich in carbon, hydrogen and oxygen, and they also have a large quantity of nitrogen, and sometimes you'll find sulfur there as well. Nitrogen is an important compound in the formation of proteins, and this really should be linked with the understanding of the nitrogen cycle. Before we go on to looking at the structure of proteins, it would be a good time to have a look at how we might test various different foods for the presence of proteins in them. And the main test that you will have come across before is Biuret's test. This starts off as a pale blue colour, and that is because it contains copper sulfate. It also has sodium hydroxide in it. And when it is mixed with anything containing proteins, the chemicals will react with the peptide bonds which are in the protein, and this results in the change of colour from a blue to a purple colour, very similar to the one on this text box. And this photograph here also shows how the Biuret's test changes. So it starts off as a blue colour and then goes quite a distinctive purple lilac colour. Looking at the structure of proteins, all proteins are made of repeating monomers called amino acids, and here is a diagram of a typical amino acid. Apart from the elements that you can see here, which are quite clear, the nitrogen, the hydrogen, the carbon and the oxygen, we also have in this square box the letter R, which stands for the residue, and this is a variable region on amino acids. There are 20 different types of R group, and each one gives the amino acid its unique character. Each amino acid also has an amino group. Here's the amino group on this molecule, and amino groups are any molecule which contains a nitrogen and hydrogens. So we've got the NH2 group here, which gives the amino acid its basic quality at this side of the molecule. We also have an acid group, and this acid is what we call a carboxylic acid, and this gives the molecule acidic properties as well as alkaline properties. So we would describe this molecule as being amphoteric, which means that it has both acid and basic qualities. Acids are defined as any molecule which donates protons or hydrogens. So the acidic end of this molecule will release its hydrogen, and the amino group, being basic, is defined as any molecule which will pick up hydrogens or pick up protons, and so the hydrogen on the acidic end of this molecule will detach itself and move across to the basic side of this molecule. It also means that proteins have the ability to act as buffers. Buffers are molecules which can react to changes of pH and moderate that change so that the pH doesn't deviate too far away from its starting point. So if we have a pH 4 buffer, then the pH 4 buffer will make sure that it remains at pH 4. So proteins have that quality, which means that it provides a slightly more stable internal environment, because changes of pH can obviously be quite damaging to proteins. So let's have a quick look at the R groups. Now R groups are very variable. If we think of the amino and the acid groups on the molecule as being in balance with each other, most amino acids would therefore be neutral. However, the R groups can vary that quite a lot. They can also change the amino acid in a number of different ways. So they could be very big, they could have a charge on them, maybe a positive charge, they could have a negative charge. They might be acidic or they might be basic, and that would make the amino acid either overall acidic or overall basic. They could be hydrophobic, so not easily soluble in water, or they could be hydrophilic. They could also include sulfur, which is quite an important one because sulfur has the ability to bond with other sulfur molecules. They could include ring structures. Now, all of these properties will affect how the amino acid behaves, and therefore it will affect the protein that is constructed from them. Now, plants and animals both need proteins in order to grow and repair. Many of their processes require proteins in order to be carried out. So if we look at plants, well, plants have to create their own proteins. So they manufacture the amino acids they need. And in order to do this, they need to absorb nitrates to produce the amino groups, which can then be bonded onto the other organic groups, and this will create their amino acids, which can then create their proteins. Animals have to take in proteins as part of their diet. 8 to 10 of the 20 amino acids are what we call essential amino acids, and that means that we have to eat them because our bodies are not capable of manufacturing them from any other amino acids. The rest of the amino acids can be made by a process called transamination, which basically means that if you've got too much of one amino acid, they can be converted into another amino acid. However, the essential amino acids have to be eaten and cannot be created from others. We can't store a great deal of amino acids because the amino group makes them quite toxic in the body. So the body has to get rid of excess amino acids by this process of deamination. And this is carried out in the liver. And the product of this is the chemical urea, which is excreted by the kidneys. So once we start with our amino acid, we need to think how we're going to create a peptide bond. So here we have a diagram showing two amino acids, and these amino acids bond to each other by something called a condensation reaction, which you've come across before, and this then releases water from that molecule, and that creates the formation of a dipeptide. And although you can't see it clearly on here, there is actually a arrow pointing to a peptide bond on the bottom molecule. So the bond between the carbon and the nitrogen is a peptide bond. Now you can actually reverse this process by a hydrolysis reaction, which involves adding back water so that the bond can be broken between the two. The dipeptide bond is a very strong bond. It's a covalent bond, and the main mistake people make with this one is that they often refer to it as a dipeptide bond. So just be careful that you correctly refer to it as a peptide bond. Now you can create proteins from amino acids. We start off by making single amino acids into dipeptides, and then these join together to form polypeptides. Eventually, if you get to over more than about 100 amino acids, they tend to be called proteins. Now, amino acids in proteins are known as residues. This comes from the R groups, and that's really because once the molecules have bonded together, the main active part of the molecule are the R groups, and therefore the most important part of the amino acid. Sometimes you're asked to draw a diagram which joins together amino acids, and I do encourage you to practice this by having several amino acids in a group, because it gets you into the swing of how the molecules are fitted together. So have a go at that first. Now, for any of you that are struggling, let me give you a little bit of help and advice. Whenever I'm asked to do this, I start off with putting down the order of the atoms that I need, and it always works with one nitrogen followed by 2 carbons and then back to nitrogen again. And so if you do it in that order to start with, that's the starting point. So you put your bonds in between them, And then you can start adding in all the rest of the things. Now, assuming you understand your amino acid structure, you can put the hydrogens in at one end to create your amino group, and at the other end we're going to have our carboxylic acid group, so we put that group in there. And if we look at the carbon directly next to the nitrogen, that's where you'll find your R groups. And then our hydrogen is on the other side of that. So we're only left now with this carbon and this nitrogen group in the middle. The carbon has a double bonded oxygen on it, and the nitrogen has a hydrogen on it. And we can also then identify the peptide bond, which is there. I've highlighted it in pink, between the carbon and the nitrogen. So whenever I draw one of these molecules, I always draw it in that order. And that would apply for a dipeptide or anything even bigger, such as this tripeptide. This would be a good point to remind you that the polypeptides and proteins are created according to the code which is kept on your DNA. So one gene will code for one polypeptide or one protein. And it's the sequence of the bases in your DNA which will determine the sequence of amino acids. Every gene is copied into a messenger RNA strand, and this then will thread onto a ribosome, and the protein synthesis will occur at the site of the ribosome. And then the messenger, according to whatever the messenger RNA states, the amino acids are assembled in the correct order, and then they're joined together by peptide bonds. You'll need to have a look back at the PowerPoint on protein synthesis to remind yourself about that, but the next slide just summarises the main points of protein synthesis. You can see here that in the nucleus we have transcription going on, where the messenger RNA is being created onto the template strand of DNA, and then this passes through the nuclear pores into the cytoplasm, where it's translated on the ribosome, and then this results in the creation of a polypeptide chain. Bonding together the amino acids produces the primary structure of the proteins. We can see in this diagram on the slide that we've got a range of different amino acids joined together. They've been given different letters, so we've got glycine and isoleucine, valine, cysteine, and so on. And so you're not expected to know these letters, although some of them may well become more familiar to you as you carry on studying the A-level course. So the primary structure of the protein is the specific sequence of amino acids that make up the protein. Now we're going to come onto this on the next PowerPoint and go through the organisation of the proteins. But let's just have a think about the possibilities and the possible different arrangements that you could get. If you had a protein chain that was 4 amino acids long, in every position there were 20 different alternatives, 20 different amino acids that could be placed there. So even working out with a protein chain of only 4 amino acids long, we have 20 times 20 times 20 times 20 possibilities. So we could create 160,000 different amino acids, different proteins from that combination. It's important to remember that to form proteins and create or break that peptide bond, enzymes are going to be required. And these enzymes are called protease enzymes. We tend to think of them as only being involved in the breakdown of proteins, but they're also important in the formation of proteins too. We can look at a couple of examples of how proteins can be broken down or why they're broken down. And the first one is hormone regulation. Quite a few of our hormones are proteins, and once the protein has circulated in the blood and the hormone has hit its receptor target, for example a glycoprotein on the cell membrane, it will have triggered a response in that cell, and then once that's over, the hormone needs to stop acting on that cell. So after it has been received, it will be broken down, and all the amino acids that are produced from that breakdown of the hormone can be recycled and used again. Aging is a particular type of breakdown of a protein called collagen. As we age, our ability to replace this collagen gets less, and so we see this in the appearance of wrinkles and the loss of elasticity of the skin, and this is due to the lack of collagen underneath the surface layers of our skin. Looking at the kind of questions that you could be asked about with this, we have a question here referring to this dipeptide, and this is being formed from two amino acids, alanine and cysteine. Using the information in figure 3.1 to help you, state how the two amino acids alanine and cysteine differ from one another when they are not combined together. So what you need to do is have a look at the molecules that you've got, refer directly to the diagram, and make sure that you use the information that you have been given. Let's have a look at the mark scheme. The mark scheme is expecting you to identify that because these are two different amino acids, they're going to have two different R groups. So if you had just mentioned that they've got different R groups, that would be your 1 mark. If you had identified the fact that one of the amino acids contains sulfur and the other one doesn't, then that would be another worthy mark. So if we just look at this molecule here, we can see at the top of the molecule we've got the two different R groups, which I've highlighted in red, and the two possible answers for your 1 mark. The next question is asking you to draw a line around the peptide bond. We'll just have a quick look at the mark scheme. The line should not extend more than halfway along the neighbouring bond. This really is stating that when you do your drawing, make sure that you're very accurate in where you put the circle, and that you don't have it covering more than you expect it to cover. So you don't actually go over other bonds as well as the one you want. So I've highlighted it here on this diagram, and if you'd drawn it like that, it would be fine, but if you ended up going over any of the other bonds nearby, it could count against you, so you need to make sure that you're fairly accurate. The last part of the question says, state what is meant by the primary structure of a protein. So this is the sequence or order of amino acids.